My name is Andrew Gordus. I am an assistant professor in the Department of Biology at Johns Hopkins University. I also have a secondary appointment in the Solomon H. Snyder Department of Neuroscience at the School of Medicine at Johns Hopkins University. I earned my Bachelor's of Science in Biochemistry at the University of California, Davis. And then I earned my PhD in Biophysics from Harvard University and my graduate work with Gavin Macbeth developing protein microarray technology. And subsequent to that, I did my postdoc with Corey Bargman at the Rockefeller University, where I did behavioral neuroscience with C. elegans, a small nematode. And then I joined Johns Hopkins University as an assistant professor in 2016. And I run a behavioral neuroscience lab where we use worms, C. elegans, and orb-eating spiders, Ulivorus diversus, as models to understand how internal state is encoded in brains.
That's very cool. I'm actually in the C. elegans lab right now, and so I'm working with them every day.
Oh, cool.
And then do you mind explaining a little bit more about your research in detail, but in terms understandable to a high schooler?
Yes. So the goal of behavioral neuroscience is to understand how behaviors are encoded in brains. However, a challenge is identifying what a behavior is. We colloquially refer to behaviors, but there are different ways you can define a behavior, right? If I reach for my mouse, I can reach for it in different ways. You could say that's reaching for the mouse, but in my brain and motor cortex, that action is encoded differently because I'm using different motor neurons. So explicitly, behavior at the most refined resolution is a coordinated activation of motor neurons. Now, of course, for complicated organisms like ourselves, that's very difficult. But with C. elegans, since they have limited neuromusculature, reliably defining behaviors in different animals is very easy. So if two animals perform this same sensorineural behavior, that's the same exact behavior. And that's part of the reason why we use C. elegans. Another reason is because we're interested in internal state. Internal state are things that we call like emotion or motivation or arousal. And those are influenced by things called neuromodulators, chemicals in the brain that alter how our brain behaves. And those neuromodulators are conserved throughout all animals. Many of those neuromodulators are things called neuropeptides. We have about 200 in our genome. C. elegans also has about 200 in their genome, which seems crazy because they only have 302 neurons. It's the only brain that's been completely mapped out and annotated. So we know exactly every single synapse, every single neuron. It's a really great system to understand how chemicals can then alter how the network performs a computation.
Oh, okay. With your methodologies, are you guys constantly imaging the worms? How are you stimulating them?
Yeah, so we use a variety of approaches. We use mutants that lack genes. We use channel rhodopsin, which is a light-gated ion channel. So we can genetically modify the worms to express this tool in specific neurons. When we shine light on them, that ion channel opens up. We can depolarize specific neurons. We can also image neurons at specific wavelengths of light by putting a tooling called GCAP. It's a protein which fluoresces in the presence of calcium. And when a neuron depolarizes, there's a massive influx of calcium. So we can use the fluorescence of the neuron, the flickering, as a measure of neuronal activity, because when it's fluorescent, that means the neuron is on, and when it goes off, it's off. So yes, we make lots of little microfluidic devices and try to understand how these neuromodulators influence behavior. So the real challenge is we've known the network for C. elegans for 40 years, right? Information isn't encoded in the structure of the network. It's important for information, but information really is encoded in the flow of activity through the network. So for example, if you took a city and you completely cloned all the roads, the information isn't encoded in the structure of the roads, it's encoded in the flow of traffic on those roads. And if you narrow a road or put in a stop sign or something, you can alter the flow of information. That really is information, the flow of the traffic. What neuromodulators do, they influence how traffic is flowing along those roads or along those synapses. So they don't alter the structure of the network, they structure how the network behaves.
Oh, I've never thought about it that way, but that's a really good analogy. And then my next question is, how did you get into research? What piqued your interest in the first place?
I don't know, I've always enjoyed puzzles and stuff. When I was an undergrad at UC Davis, I saw a flyer to do research on a plant lab. And so it was an NSF fellowship, and I got the NSF fellowship to do research. And I joined this plant lab, and the lab was trying to understand how proteins are transported into chloroplasts. Chloroplasts are these little organelles that harvest sunlight to produce chemical energy. I really enjoyed that. So I thought, well, I'd go to grad school and learn how to do more research. I was really interested in tech development when I was in grad school. So I focused my research on developing biotechnology. But then when I was in grad school, I got interested in how information is encoded in networks. So a lot of my tool building was to try to build tools to understand how cell signaling networks encode information. Because the challenging thing about biology is that everything you do is a result of chemistry, right? But chemistry is inherently entropic. It's very noisy, and yet I can do all these ordered things. That's because evolution has evolved all these signaling topologies, these sort of tools that really decrease the contribution of entropy to signaling. And so you get these defined signaling outputs. And I was really interested in those principles and how they apply to the brain. And so I started doing a neuroscience journal club. I learned more about neuroscience and I applied to a neuroscience lab after my PhD to learn more about it and to try to pursue these questions in a very defined, simple system.
Yeah, and then this kind of leads to my next question, which is I wanted to ask what prompted the change from biochemistry to biophysics? Because I feel like those seem like separate fields.
Oh, yeah. I mean, everything in biology is very overlapping. During my biochemistry major, I took a lot of physics courses. I was really interested in the fundamental principles that govern how biochemistry works. Like in my undergraduate lab, I worked on the kinetics of protein transport, and I fit kinetic models to the data. So I've always been interested in more like quantitative methods. So when I did my biophysics PhD, I took a lot of graduate courses in physics, developed a really sort of refined understanding of how quantify things and tool development technology. And I applied that in my postdoc, and I apply that now in my lab. I do a lot of engineering of tools, especially with the spider research, where you do a lot of building of things and writing of computer algorithms.
Oh, okay, that makes sense. Like, where do you see your research field expanding to in the future? And like, what do you think students can focus their attention to?
When I got here, I expanded my research into spiders. I got interested in spiders because a real challenge in behavioral neuroscience when it comes to these internal states I mentioned, like arousal, motivation, emotion, stuff like that. You know, all animals have internal states. I hesitate to use terms like emotion to invertebrates, but invertebrates also have arousal states to influence their behavior, like satiety, sexual arousal, conspecific arousal, stuff like that. And a challenge, though, is that you can't ask an animal how they feel. Okay, I can ask how you feel. That you can provide all sorts of words, right? Even with humans, like how many internal states are there? You can say you're excited, but there's different types of excitement, right? Different types of hunger, different types of fear. All those are influenced by all these neuromodulators. Ultimately, all those states are influenced by different combinations of these neuromodulators. Now, with you, at least, I can get some refined definition of like how you feel based on asking you questions, right? But I can't ask an animal questions. What I have to do is watch an animal and try to infer their state based on their behavior. For example, a cat, when it's content, it licks its paw, right? You've seen a cat lick its paw. It's like, oh, that's a very content cat. But actually, cats lick their paws all the time. It's the probability of licking the paw, the frequency of licking the paw that really defines whether they're content or not. If you just see them lick their paw once, maybe they're content, maybe they just had something in their paw. But if they're sitting there really going to town on their paw, you're going, okay, well, pretty likely the cat is content. The challenge is inference based on statistics. The nice thing about animal architecture is that they're providing you with a physical record of their intent. Many animal architecture go through phases. So they're telling you, like, I am focused on building this, and now I'm focused on building this. So it's a really nice system because they're providing you with a physical record of like what they're trying to do. I don't have to ask them, they're showing me. And so we got interested in orb weavers because the orb web, the classic spiral web, like kindergartners draw, that's a really nice geometric structure that's easy to quantify. Like it's like bird nests and stuff. Like I don't even know how to quantify a bird's nest. It's very complicated, right? Also, birds have to constantly scavenge for things to build their nest. The spider is like producing material out the end of their abdomen, right? It's all a very sort of confined problem. And I like sort of nice confined problems. So when I came here, I expanded into spiders because I think they're a nice system to understand internal state. The direction of the lab, just trying to understand like what are internal states, how are they encoded by neuromodulators, how do those neuromodulators alter the structure of networks?
Yeah, that's really cool. And spider is something I haven't really heard before. Like I feel like it's a very unconventional model.
Yes, back in the 70s and early 80s, a diversity of animals were used for behavioral neuroscience, and they're still used. But there was a real consolidation of research into model organisms like Drosophila, fruit flies, C. elegans, mice, stuff like that, because all these genetic tools were developed in the 80s and 90s that really made them great genetic systems. However, they're really limited as far as behavior. They only perform a limited number of behaviors, right? If you look across the planet, so many crazy behaviors have evolved. Like I would love to research all of them, but the animals weren't as tractable as a system. However, recently a variety of genetic tools have developed that are generally applicable to more organisms, like CRISPR, for example. A lot of engineering tools have been developed. A lot of computational tools, especially machine vision, have been developed to really enable us to develop behavior models with really refined spatial temporal resolution, because these machine vision algorithms allow the computer to sit there and annotate the the behaviors rather than having us sit there for hours on end. And so we can sort of build more robust statistical models with this machine vision.
And then my next few questions are more geared towards high schoolers. How did you find your passion for research, and how do you suggest others find their passion? What would draw someone into research?
Just being curious about the world around you. I grew up just south of Yosemite National Park. Oakhurst is a small town at the south gate of Yosemite. And I grew up sort of around nature. I really enjoyed being out in nature. I did lots of hiking and rock climbing and hunting and fishing and all sorts of things. And I think that combined with my curiosity. I'm just always curious to know how things work. Every morning I do a Wordle and a connections, puzzle. I do crosswords. I just love puzzles. And so I see life around me as a big puzzle to solve. And that's really what drives me.
I do connections in Wordle every day too, and I feel like it's just like a built-in like habit now. I really enjoy it. Like I'm sure you get a lot of cold emails and like requests to join your lab from, a variety of people. How do you pick out someone? Like what makes someone special or someone stand out to you?
I mean, there's different levels of people who reach out to me. There are high schoolers who reach out to me. I can only have one high schooler at a time. So I'm partial to Baltimore public school students because I'm part of this program. And so when it comes to them, if a student really shows a genuine interest and they've gone through and done background research on me, I think that shows a genuine interest in the work, because training high schoolers is so much work. If they're not genuinely interested, then it's going to be a real uphill battle. If like they're just trying to check a box, it's a real drag. So I look for people who are genuinely interested. That's true for undergrads as well. I can have more undergrads. They're a little easier. Typically I have about 6 at a time. And if they show a genuine interest, they come talk to me, they ask questions. Really just curiosity and an interest is really what drives me to pick people. I don't really care so much about prior experience or skills, because I see high schoolers and undergrads as basically having no skills, and my role is to provide them some training. As I move up to graduate students and postdocs, I start to consider skills more seriously. Now that's just me. I know other faculty look into skills and stuff, but I feel like that's kind of biasing at that level, biasing towards students who have had a lot more opportunities in their lives, And so for equity reasons, everyone can have, if a student has a lot of genuine interest and curiosity, but have had very little opportunities in their lives, I will typically choose that student over someone who has a lot of opportunity, but does not seem that curious or interested.
Yeah, I really appreciate professors who do that, because I feel like as a high schooler, if there's no opportunities, people won't take me because I don't have prior experience. I can't get prior experience if, there is no place to start.
Exactly. It's like you're stuck in this catch-22. So I just care more about genuine interest.
Yeah, and then my last question for the day is, what traits make a good researcher? Like what makes someone that you want to work with or someone you want to mentor?
Curiosity and a drive to answer your questions. Like I've had students who are curious, but not invested enough to invest the time and mental energy into answering the questions. You need to have the curiosity and the drive to answer. Those are like the really 2 key things.
Yeah,
because
definitely.
Those 2 qualities, if you have that, you will just naturally acquire the knowledge needed to answer your questions, and you will acquire the skills needed to answer those questions because you're driven to answer them. So for grad students, I don't care about whether they have prior knowledge. If I had a student who knew nothing, she turned out to be a great researcher because she was very driven.
Yeah, everything is like a byproduct of this too. That's all the questions I have for today. Thank you so much for your time, and I really enjoyed talking to you.